Hello, this is Mrs. Murdock, and we are doing a makeshift review of Unit 6, right? That's what we're doing. So what we're going to do is we're going to differentiate between prokaryotes and eukaryotes, first of all in terms of their basic structures, then in terms of the DNA, and then in terms of protein synthesis. Okay, let's do that. So first of all, tell me everything you can about prokaryotes versus eukaryotes. What can we come up with?
Circular DNA.
Very good. Yes. The DNA is circular here. Very good. And here it's linear. Exactly. It's linear DNA here. And here it's in the cytoplasm, right? It's free in the cytoplasm, and that becomes significant during protein synthesis. And here it's inside a nucleus, which makes a really big difference when you get to protein synthesis and transcription, translation being separate processes, right? Okay. So circular DNA, all that kind of thing. Okay. So now let's talk about transcription. Transcription for both of them, right? So transcription in a prokaryote, how is DNA organized in terms of the genes along the string of the DNA in a bacteria? It's called a what? It's called a operon. Very good. Okay, transcription depends on repressors, right? And inducers that interact with operons, right? Operons are a prokaryotic phenomenon, okay? And over here with the eukaryotes, it's a very different thing. Transcription over here. In eukaryotes, depends on presence of transcription factors. Remember those? Transcription factors, which are usually small organic molecules, often proteins, right? That's what they are, that interact with promoter regions that are upstream from single genes, right? That are upstream from single, not multiple like with operons, but single genes, from single genes. Running out of room down there. There we go. Okay. Everybody with me so far? Okay. So there is some differentiation there, right? So we've got these transcription factors that have to be there at the promoter region. Remember, operons also have promoter regions, right? But in terms of bacteria or prokaryote, right, you might have several structural genes here. You might have X, Y, Z genes, several of them, with upstream a single promoter and maybe an operator here, and regulatory genes right here that may be making things like repressors and things like that, right? Repressors come from regulatory genes, or you might even have an activator that comes from another one, like the lac operon, which makes CAP. Remember that? So with a eukaryotic gene, you have some things that are in common with that, but it is very different. So if I do the same sort of thing with a, where's my thing? If I do the same sort of thing for a eukaryote, oops, sorry about that. For a eukaryote, you only have one gene, Only one per promoter. Like here's, if this is your promoter region here, that's it. And you've got one gene here and only one. Whereas an operon has a promoter too. There's a promoter here, but it might be in control of several genes that have one sort of process in common. Like the lac operon has all the genes that you need to let lactose in and then digest it, right? And you need several genes to do that. But with eukaryotic genes, it's one promoter, one gene. One promoter, one gene, right? Okay. I don't know what that's doing there. I'm just going to keep going. Okay, so if I keep going down here. All right, and then transcription factors, you know, would kind of connect on here. And sometimes, like with the video that you watched, there's a little bit more to it. Sometimes this comes over and bends over and there's enhancers involved like that. But you can go look that up yourself. Okay. All right. So then let's keep going. Let's differentiate a little bit more here, right? So with prokaryotic genes, once you have the RNA polymerase here, right? Once you have the RNA polymerase enzyme there, and transcription starts, can you stop it? Can you edit the RNA? Can you do any kind of interspersal regulation there? Nope, you can't. Once a bacteria cell gets going, it's going to go all the way to the protein, right? Once transcription starts, these are going to go all the way to proteins. They're going to get translated. You're going to get your messenger RNA, right? You still do that, right? But transcription and translation happen very quickly in a bacteria. In fact, as soon as the RNA polymerase sends out that messenger RNA, the ribosomes are going to come up to it immediately and get started translating it before it's even finished transcribing. One of your mastery questions was that, right? Your mastery question said one of the big differences between eukaryotes and prokaryotes is that prokaryotes can do their transcription and translation at the same time. Okay, and definitely you can't do that in a eukaryote, right? So let's say that you have RNA polymerase binds here successfully. Let's say that that went well, the transcription factors were there, the chromatin was unwound enough, all of those things were in place, and transcription starts. So then what happens next? You get a messenger RNA, right? You get a messenger RNA. And then what happens before translation? Okay. So here's the thing, another huge difference between the prokaryotic genes and the eukaryotic genes is that these guys have these introns in them, right? Built into the code. I'm making little eyes there. There's introns in a eukaryotic gene. And then what are the spaces in between? Those are exons. Very good. So the exon, there's a couple of them here. Right? Exon here, exon there. Right? Okay. And then when the messenger RNA is made, you've got those same introns are still there. It's not like they go away. They're still there. Right? We've got some 1, 2, 3, 4. I made 4 there. I'll make 4 here as well. There we go. So I've got 4 little introns there. What's going to happen to them when I edit? Yeah, so we have editing happening or processing. They call it both of those things. Sometimes they call it RNA processing, or sometimes they call it editing. And what you're doing is you're splicing out the introns. You're splicing out the introns. And that's something that the prokaryotes cannot do. So if I go over here, there's no messenger RNA editing, right? That's what we said. It goes straight to the proteins. The protein products are made with no editing, right? Whether over here, the final messenger RNA is going to be a little shorter, right? Because it'll have the introns out, and you'll have the exons spliced together. So there are 1, 2, 3, 4, 5 exons that'll be all put together. 3, 4, 5. Okay. With the introns spliced out. Good way to remember this in terms of the language. The exons leave the nucleus, right, to go get translated. The introns stay in the nucleus and get recycled. Does that help? Yeah. Okay. So then there's something else that they put over here on the end. What do they add to the end over here? On the, yeah. They add a polyadenine tail. Adenine, sorry. A poly-A tail. In other words, they add a bunch of A nucleotides. Adenine nucleotides, right? And over here, they put a methyl cap. A methylated cap over here. And supposedly, I think this actually helps it connect to the ribosome when it gets to the cytoplasm. And the adenine tail is supposedly, when you get into the cytoplasm is a dangerous place. So there's a lot of enzymes that are going to go after the end of the tail of the messenger RNA and digest it, right? Because the messenger RNA only has a certain lifespan to be translated. So the poly-A tail gives them something to eat without getting into the genetic code of the gene. Okay. So then this final thing goes into the nucleus, right? Goes to, whoops, to nucleus for what process? Translation. Part 2, translation. All right. So that involves the ribosome, right? You know that. And translation, in terms of both of them, the two processes in the two different kinds of cell, I'm going to say, are really pretty similar. The only difference between translation in a bacteria and translation in one of our eukaryotic cells is the location. Over here, we're in the cytoplasm, right? Here, we're in the cytoplasm too, but can be simultaneous. Is it okay for me to use that big SAT word? Simultaneous with transcription. That may be really the only difference. Did I say go to nucleus? I'm sorry. I'm not even thinking straight. Goes to cytoplasm. Sorry, I'm a little bit tired. Okay, there we go. You guys knew what I meant. Oh, thank you. Okay. Okay. But in terms of that, there's not really a difference. They both need ribosomes. Remember, bacteria have ribosomes too. They're not membrane bound, so the bacteria are allowed to have those, right? Even though you're thinking, oh no, don't prokaryotes have no organelles at all? They have ribosomes. Ribosomes evolved way, way, way back there. Even the mitochondria that we talked about in class also even have ribosomes. So there you go. Okay, other than that, maybe something I can add up here that I think Megan was asking about is that whole 3 to 5, 5 to 3 thing, right? So if you think of it this way, whether you are a DNA polymerase or an RNA polymerase, what way are you reading the DNA template? What direction? Always 3 to 5. Right. Both polymerases will read in a 3 to 5 direction on the template, but then they'll create a new molecule in a 5 to 3 direction. So if I put, if I put a 3 here and a 5 here, that's right, isn't it? Then the RNA poly is moving in a 3 to 5 direction along that gene. Same here. The prokaryotes do the same thing. 3 here, 5 here, right? So that's correct. Oh, about to run out of time. Okay, that's it for the video, but I'll keep talking, okay? I hope so much so far this has been helpful.